Dear Professor Hong and distinguished researchers of KIOST, thank you for the invitation for the Marine Science Institute scientists to give their presentations. I speak on behalf of the Marine Biotechnology Group of the MSI. This is the title of my talk, and I would like to say that because there is very little time, we would probably have to breeze through some of the slides that were provided by my colleagues. So which one should I use? Oh, okay. So the challenge in marine biotechnology is to make use of marine resources, including those that are ravaged, those that are threatened by anthropogenic factors, and also to make use of marine organisms that seem to have harmful effects on humans. The paradigm being that in a complex, intimate environment like marine oceans, coral reefs, you have the opportunity to convert harmful effects to beneficial effects, depending on the level of science that you will undertake to understand what we know today as the biomolecular interactions and mechanisms of action among these marine organisms and the physico-chemical environment. So these are our ecosystems and resources that the MSI is interested in, and our holistic approach is to study the biology of the organisms as well as their ecology, and use chemistry and biochemistry as the underlying strategy for understanding biology and ecology, to find useful applications as well as to make this sustainable through biodiversity, conservation, and development. So we focus on these approaches, and in the photos you will see a major interest in a mega diverse group of marine organisms in the Philippines. These are the mollusks, and it is because of geological events that we have in central Philippines, in the Visayas region, the highest molluscan biodiversity in the world. Okay. So here we have our curriculum for the Marine Biotechnology program, and you will see that it is interdisciplinary. We require our students to know ocean physics and chemistry and also biological oceanography. But we bring down the level of understanding to the molecular level. Also note that the program is research intensive. It is flexible. We can have master's students, we have PhD students who will pursue the degree program straight without a master's but with publications, and also those with masters can pursue their PhDs with different curricular requirements. So these are some of the electives that we have, and I teach and team teach some of these courses, and it's a combination of lectures as well as laboratory courses. I'd like to start by featuring and honoring our mentor at the Marine Science Institute. She is a woman. She is a professor emeritus and national scientist of our country, and she was awarded L'Oreal Women in Science Laureate Award for her work on conopeptides that I was referring to as a mega diverse group in our country. And in collaboration with another Filipino scientist based in the United States, they published in the mid-80s work on a conopeptide that is produced by a conus snail in its venom as part of its prey hunting strategy. A peptide that is a drug today known as pre-alt, which stands for primary alternative to morphine. Its other name is ziconotide, and it is an anti-pain agent. So all of us are mentored by Dr. Lourdes Cruz. Today, she collaborates with Professor Rodora Asanza, who will speak tomorrow, today also, on harmful algal bloom toxin analysis and diagnostics. And she's also my antecedent or predecessor in the conopeptide work that I continue to pursue with her collaborator, Baldomero Oliveira of the University of Utah. So I shall start by talking about my work. So one of my projects is on the conopeptides, which is actually a toxin, but a neurotoxin can become a neurodrug if you administer very low doses of it, and if you know what the neuropharmacological or muscular target of that peptide is. But before that, I will talk about a program where I am associate program leader, and it is to study the bacterial symbionts of these conoideans, as well as another group of mollusks called shipworms. The rest of my projects are marine-related, but they are also biomedical projects. The third project that I will tell you a bit about is the synergistic studies that I'm doing on marine compounds in combination with clinical drugs for cancer. The idea being that nature's treasure trove of drugs should not be depleted by administering these drugs singly, because it's just a matter of time before a cancer cell or an infectious microorganism like Staph aureus will develop resistance to that single drug. So today, the strategy in drug use is combination therapy. And this is what we are advocating at MSI and in my research group. So let me tell you about this PMS-ICBG research program funded by the US National Institutes of Health, Fogarty International. And I collaborate with the University of Utah. Our principal investigator is probably known to some of you. She is the molecular microbiologist Margot Haygood, formerly with UC San Diego, then with Oregon, OSU. She's transferred to the University of Utah. And her right-hand person would be Eric Schmidt, very famous bright star of marine natural products. So I am the associate program leader based in the Marine Science Institute, where we do our sample collection, our bioactivity testing, and also chemistry of the natural products. In another project of that program, we have Dan Distel of Northeastern University in Boston. He's the world's expert on shipworms. They look like worms, but they are actually mollusks. And tying us up all together is the famous biochemist, Filipino biochemist Baldomero Oliveira, who is known for his work on conopeptides. So here are some compounds that we have isolated, and this group of compounds, they're modified peptides, and some of them are cyclic. They were isolated from bacteria that live in the mollusk Chacorius nobilis, and also in Conus stribliae. So we call them nobilamides, and we are able to show that they have significant binding activity to an important molecular target in pain, TRPV1 receptors or channels, which are rich in dorsal root ganglion neurons, which some of you might know, well, are found in our spine area, and they're the ones that mediate your sensations of heat, touch, pain, etc. Okay? So very important pain or sensory target. And what we do is to culture DRGs, or dorsal root ganglion neurons, from mice in the laboratory, both in Utah and in my laboratory. And we are able to show that some of these extracts and compounds can change the effect of a compound like capsaicin, which is rich in kimchi and in other Korean food products. Okay? Therefore, these compounds are potential anti-pain agents. Why? Because capsaicin or kimchi initially has this stinging effect, but the reason Koreans like it is because after some time it has a numbing effect. So you can take a lot of the kimchi and you can enjoy the other benefits of whatever you kimchied, the veggies that you kimchied, for your health and wellness. Okay? So we are aware that Korean food has evolved this way, that you have many varied ingredients that are very healthy, including the very colored and stinging or spicy ones. So that's the nobilamides. Then let's move to this group of compounds, the polycatins. As I said, we have an interest in neuroactive compounds. And the reason for this is because the snails, where we found these bacteria, have neuroactive peptides in their venom. And that is the way they paralyze their prey. They sting a fast-moving fish to paralyze it, or a fellow mollusk, or a worm to paralyze it and to eat it. The snail also uses compounds, it's becoming very clear to us, to deter their predators and their competitors. So it's no longer just for prey hunting, it is also for deterrence of their own predators. Even if they have a hard shell, they have predators. Imagine if a lobster came near them, they'd probably lose out. Okay, competitors and predators. So let me now go to the polycatins. So these compounds, which are thiazolin compounds, they were isolated from bacteria that are symbionts of Conus polycarius, thus the name polycatins. And we had this regimen of testing them, first in mice. We have mouse testing following our own ethics committee's rules. Then we had them tested in the DRG assay on neurons that we cultured in the lab. And then when we had this nice group of compounds, then we sent them to North Carolina, because at the RTI, in one of the universities there, they have the Center for testing about 40 or 50 neuromuscular targets. And they were able to show that the polycatins are selective 5-HT2B receptor antagonists. And this receptor is important or related to memory and learning. So it's got a neurological function. I'll move on to the nocapyrones. The nocapyrones, these are polyketides. they have been reported in the literature, but we were able to isolate and identify new members or analogs of this group of compounds. And the interesting thing is, through instrumentation, through mass spectrometry, we were able to show that the mucus of the snail, from which these compounds are isolated, had a significant amount of the compound. Okay? So that establishes the finding, the position that perhaps these kinds of compounds have an ecological and therefore a biological role in perpetuating the snail that is hosting the microorganism that produced these compounds. So in all these cases, what we did was to do conventional microbiology, isolate microorganisms from the different tissues of one snail, grow them in the lab, select, purify interesting microbial isolates, culture them in broth in large quantities like liters, and identify the compounds, purify them, and test them for biological activity. It's a very difficult process, and it's like looking for a needle in a haystack. Okay, but we have published this in the literature, and we continue to work on our microbial isolates because they have yielded very promising results. I'll shift now to the work that I'm doing on another group of compounds isolated from sponges. So my original area of research was sponges. So I have publications with Chris Ireland from the University of Utah on compounds from marine sponges. And we say that sponges are very important because they are the first multicellular animals. And so from prokaryotes to eukaryotes, you have the sponges as, landmark organisms. Clearly, something happened to them so that cells could now aggregate into multi-cells. So this is an important model, actually, for human tissues, organs, evolution, but also an important model for cancer, because we know that cancer has unique properties that puts it at advantage over normal cells. And one of this is the ability to aggregate and to propagate, to invade and to metastasize. And so if you look at the sponge genome and the human genome, you'd be surprised that there is similarity in their genomes. And for that purpose, we firmly believe that sponges have genes, have biosynthetic capabilities to produce a whole range of small molecules called secondary metabolites that may regulate what we call cell proliferation and aggregation. So here we have a compound, and those in this field would recognize the raniera-mycins, largely worked on by the group of Naoki Saito of Meiji Pharmaceutical University, whom I collaborated with. And the raniera-mycins are isotetrahydroquinolines related to a drug, cancer drug, called ectane acidin 743 or trabectin, Yondelis. This is a drug approved in Europe, first for soft tissue sarcomas, and now the applications are being expanded. But as I said earlier, we do not want to just use single drugs. So my in-house RA did his master's thesis on the combination synergy of raniera-mycin with a widely used anti-cancer drug, a DNA intercalator and topoisomerase 2 inhibitor known as doxorubicin or adriamycin. And doxorubicin is affordable, but it is very cytotoxic, and it has a glycoside. So if you have a heart condition and you have cancer, then you are at risk for, heart problems because cardiac glycosides affect the heart. So here we show through a series of experiments that raniera-myosin M at very, very low doses, nanomolar doses, can improve the effects of doxorubicin, can reduce the dose at which doxorubicin can kill cells. So they act in synergy with each other. And the collateral benefit of that is, first, you don't have to synthesize, pay for as much of the drug because it's active at low doses. Second, you are not going to administer it in your circulation at a high dose that will also kill the normal cells. So you will have less of the toxicity. So this is the reason we are pursuing this kind of research. Okay, so we have several instruments in our lab that can monitor what happens to these cancer cells while they're being treated with doxorubicin alone, raniera-myosin alone, and the two in combination, at different combinations. And these are real-time cell analysis, okay? So that's the first result there. And we will not go into the details of this. And we are also looking at the cell cycle or the apoptotic effects of our combination of raniera-myosin and doxorubicin using an instrument called flow cytometry. So flow cytometry is now like a workhorse instrument, and it is also used in the field for marine cellular studies. So that ends the first part of my talk on marine drug discovery and development. This is work also under the US NIH ICBG with Dan Distel, Margot Haygood, but one of our bright stars in my laboratory, Marvin Altamia, was the guy who isolated this microorganism from this mollusk that looks like a worm. It is the biggest shipworm ever. It's this long. And it's a magnificently ugly, gooey creature. It's got a calcareous shell that's color white, and it's got holes or breaks, cracks. But then inside, you can cut open the shell, and this blackish gray creature will come out with a degenerate shell. It's a bivalve. What's interesting about this shipworm is that we isolated it, or we collected it from muddy, anoxic, hydrogen sulfide-rich water in a lake where a lot of wood had fallen because there was a landslide and there was like a forest nearby and there was a logging industry nearby. So these organisms evolved so that their bacteria could make use of alternative chemicals to produce energy in the absence of oxygen. So we found in the genes of the bacteria found in their gills, rubisco, this enzyme that is complex, is very important. And so now this paper will be, this work will be published in the Proceedings of the National Academy of Science, and it is a follow-through of a hypothesis that Dan Distel had 10 years ago. It's the wooden step hypothesis. And what it's telling us is evolutionary biology is largely influenced by physico-chemical environment, or you might say oceanography, okay, or mangroveography. Because then here we saw that these shipworms, which normally harbor cellulitic bacteria in their gills, and that is why they're called shipworms, because they feed on wood in the mangrove. And they're famous because they changed the tide of history. It is the main reason, we are told, that the Spanish Armada lost to the British fleets during the time of Queen Elizabeth. That is because the Spanish Armada ships had holes due to shipworms. Okay, so we have isolated little shipworms from different parts of the country, and they contain cellulitic bacteria. Now comes this huge shipworm found in a unique physico-chemical geographic environment, and it has rubisco. It's got hydrogen sulfide, sulfur oxidizing enzymes. Okay, so we're saying that this could serve as a link between hypoxic, anoxic environments, including geothermal vents, deep geothermal vents, then those that are based in the mangroves, they're boring into wood, and so there's also limited oxygen there, then all the way to the oxygen-rich environment. So clearly, we are trying to address the problem of, understanding evolution this way, but at the same time, we're also thinking of conservation measures and also biotechnological applications, because in industry, we know that there are many chemical reactions or catalytic reactions that would have to be performed under low oxygen. Okay, so now this is a bit of diversion of how we do drug discovery all over the world, including in the Marine Science Institute. I'm now shifting to my work on the peptides found in the venom of conus snails. No longer the bacteria that are their symbionts, but the cone snails themselves. So there are about 600 species of conus snails. But the conus snails belong to the Conoidean superfamily, and they have as their older relatives the turids, which are tinier than they and live deeper in the ocean. And we are also studying the turids. There are 12,000 species of them. And they are all converged in the central Visayas region of the Philippines. Again, due to geographic events that occurred leading to the formation of the Philippine Islands. So here we are looking at how we can discover new neuroactive peptides in a different way, or in a way that will help us with our biochemistry work. Because traditionally or conventionally, we isolate the conopeptides through high-performance liquid chromatography, through chemistry and biochemistry techniques. But now, because these conopeptides are direct gene products, they are translated from their mRNA sequences, which are encoded by the DNA or the genome. So we have what we call genomics as well as transcriptomic studies, and the transcriptomes, for which we have libraries for each of the cones, help us predict, help us complete the sequences of the peptides that we are isolating in the lab. And it's really revolutionary, the way transcriptomics and genomics can speed up our research. Hand in hand with this, we are able to now chemically synthesize these peptides using our peptide synthesizers and sequencers. And so we're able to produce milligram quantities of these peptides that would allow us to test them in animals and eventually in humans. So in my lab, aside from isolating the peptides, we are developing drug delivery systems and using the chemically synthesized peptides from the Institute of Chemistry, care of my collaborator, Dr. Aaron Villaraza. Okay, so these are the sequences of the peptides, amino acid sequences, and they are classified based on superfamilies, and they have a very unique structure, 3D compact structure, because they have cysteine disulfide linkages. So we have different cysteine frameworks for different types of peptides. And they directly interact with ion channels and other neuroreceptors. Okay. So this is the work of our PhD graduate, and she's now doing a postdoc elsewhere. And we went down to the bottom line, which is to identify 3 specimens of each species of 2 species, and we did transcriptome analysis of them, just to show the similarity or divergence of their transcriptomes through the superfamilies. The person that I collaborate with on the genomics and transcriptomics is Arturo Luisma, who was the advisor of our PhD student, and I was a co-advisor. Okay, now I have presented our mentor, that is Lourdes Cruz, and then we're researchers in MSI. We recruited a young researcher from the University of Florida who was also working in my lab before she did her PhD. This is Lilibeth Salvador Reyes, and she worked in the University of Florida under Recluse, another famous person in the marine natural products field. And she's moving the field forward for the time when I retire, and she will take over the whole marine natural products research group. And her approach is immediately to combine phylogenetic analysis, okay, with HPLC or LC mass spec. Okay, so it's genetics, so that's biodiversity, and then LC mass spec, that is chemistry, functional chemistry, and then the third is bioactivity or functional, so bioactivity data. And her field of interest is cyanobacteria, and those in the field of marine natural products know that cyanobacteria have been among the most prolific producers of new compounds that are bioactive. They are modified peptides. So this is work that she had published with her mentor, Henrik Lusch, in 2015. And it identifies phylogenetically which are the species that are producing these very promising marker compounds of cyanobacteria, dolastatin, simplostatin, and largazole, which are in advanced stages of development. The dolastatins have, one of them is already a drug also. Then Lilibeth, together with Roy Luisma, they are now looking at the proteins or the group of proteins that are being expressed in the harmful algal blooms, and they are working with Professor Rodora Azanza. Okay, so there are many things that have to be understood, not just on the level of genomics and transcriptomics, but also proteomics. So perhaps with KIOST, we could move forward this kind of research with your advanced instrumentation and techniques. I'll move a bit to another field of research, which is seaweed chemistry. And Nemesio Coke Montaño recently retired, but he was our anchor person in seaweed research. And this is where I think we can also explore opportunities with KIOST, because you consider sargassum to be a menace. It kills the fish. But why does it kill the fish? You say it might be because of the bulk. Okay, it traps nutrients. It encroaches on the space. But we must know that sargassum is so successful in the environment because it's aggressive, because it produces compounds that kill others and maybe regulate its own growth, and therefore we can harness them for various applications, including cancer prevention and anti-cancer activities. So I would be very interested to study your invasive sargassum species and compare it with our species. This has been one of the advocacies of Dr. Montaño. Okay. So beyond isolating the polysaccharides from the seaweeds and the small molecules, he's moved things forward. Okay, he's moved things forward by looking at the applications. I think I will have to stop here because, well, we're running out of time, and the other topics may be covered by other speakers because our institute is very interdisciplinary, and they work with our other faculty who are speaking here today. So the rest of the projects here have to do with ecology and conservation. Thank you very much.